Hi everyone, my name is Ali Adahar. Welcome to my bacteria transformation slides relating to all units in AP Bio. I'm going to be talking about how the Let It Glow lab relates to all units in AP Bio, starting with Unit 1. This lab relates to topic 1.4, in which we learn that the cell membrane is a phospholipid bilayer. The phosphate heads are hydrophilic and the fatty acid tails are hydrophobic. Hydrophilic substances that diffuse in and out of the cell usually use facilitated diffusion or a different type of diffusion besides straight through the membrane because they can't due to the fact that they're hydrophilic and they would get stuck in the hydrophobic portion in the middle. And the heat shock makes the lipids in the bilayer less densely packed and loose, so there's holes, and therefore the membrane is temporarily permeable and the hydrophilic substances can slip through. This includes plasmids, and there's a plasmid uptake in the lab. The photo I included shows the hydrophilic head and the hydrophobic tail of the phospholipid bilayer. Moving on to Unit 2, the lab relates to topic 2.6. Passive transport is when a substance moves from an area of high concentration to an area of low concentration. After the heat shock, the plasmids move through the semi-permeable membrane from high concentration outside of bacteria cells to low concentration inside the bacteria cells. The picture I chose illustrates an example of passive transport. CO2 is moving from a high concentration to a low concentration. Next is Unit 3, and the lab relates to topic 3.3, where we learned about endothermic and exothermic reactions. Exothermic reactions are when heat is released, whereas endothermic reactions are when heat is absorbed. The heat shock in the lab is an endothermic reaction because the bacteria is stricken with heat and absorbs the heat instead of releasing heat, which would be exothermic. The Let It Glow lab relates to Unit 4 and Topic 4.7, where we learn about cell division. Cell division promotes the growth of a cell population. The bacteria samples were left overnight, and over this time period, the bacteria cells underwent cell division through the process of binary fission, resulting in growth of the population. Unit 5 relates to the lab specifically through Topic 5.5, in which we learn about gene expression. DNA carries an organism's genes, which can each be activated to express a certain phenotype. In the lab, the GFP gene is activated by lactose to express the EGFP fluorescence, which is the phenotype in which the bacteria glows under blue light. The picture I included shows a chromosome and highlights a specific gene in the chromosome. The lab, of course, relates to Unit 6 as well, in Topic 6.1. Prokaryotes contain plasmids, which are small and circular DNA molecules. In the lab, 2 out of 4 lanes in the glow tray have plasmid exposure, while the other 2 do not. The plasmids can enter bacteria cells, while the membrane is semi-permeable after the heat shock. The picture displays plasmids in bacterial DNA. Unit 7 relates to the lab in Topic 7.2. Allopatric speciation is when a physical barrier divides populations. In the lab, allopatric speciation is demonstrated because the bacteria populations are separated by the plastic barriers in the glow tray. The populations are geographically isolated in a sense, preventing gene flow between the bacteria. Lastly, in Unit 8, the lab relates to Topic 8.1, in which we learn about how organism responses to the environment can vary based on stimuli and resources. In the lab, the bacteria react differently in the presence versus the absence of ampicillin. When ampicillin is present, the bacteria develops a resistance to antibiotics. The absence of ampicillin means the bacteria does not develop this immunity. Therefore, bacteria exposed to antibiotics will survive if they have that immunity from the ampicillin, and they will die if they do not. The picture I chose displays the stimulus, organism, and response chain reaction.